Hello everyone, welcome back to another study class of regeneration of Hydra, and today we will try to understand about the mechanisms of regeneration in Hydra. So mechanisms basically means how this individual or this organism is orchestrating various gene expression events that is leading to a restored body part. So cellular and structural changes during the regeneration in Hydra is seen in this picture. As you can see here in the left extreme, before amputation, there is a head region and also the tentacles you can see. And the scissors shows there is the amputation plane. And soon after amputation, what you see is there is a retraction of this blue colored extracellular matrix or the so-called mesoglial structure which is been withdrawn back, and you could see the cells which were elongated. This is endodermal cells, light yellow, and the brown colored is the ectodermal cells. So the endodermal cells got spherical in shape, and then after around 1 hour, what do you see is these spherical cells, because the beauty of this sphere is it can move on to any direction. So it will allow it just like a football. So it formed a dome shaped structure and it merged with, and so this is nothing but a wound healing. It healed the wound. And now you can see after some time it started becoming elongate again, the endoderm cells. So if you look at ultrastructural level, this is what you see. At 1 to 4 hours, the cells get rounded in shape, and then it start around 4 to 7 hours, it start producing this matrix protein. And after around 7 to 20 hours, it started filling them with interstitial matrix. As you can see, the key in the left hand side, interstitial matrix, that is fibrillar collagen, fills it up. And basal lamina, which is nothing but the cells secreted by this epithelial and endothelial cells. And these ectodermal cells are brown in color and endodermal cells are light brown in color. So by around 7 to 20 hours, it restored the normalcy. So now what is left? It has to make the tentacles back. This is what is needed for the head to form. So ECM is retracted. That is very important. It is retracted immediately so that it gives a freedom for the cells to move around, because there is no matrix means it is free for all. It is just like you had a string of beads, a garland or a chain you had which had so many beads, and you suddenly pull the beads out of the string. What will happen? It will go randomly. So this is what happens. The basal lamina is retracted upon decapitation, so followed by change in the endodermal cell shape. And then comes, this is succeeded by the wound closure and the secretion of ECM component. The moment the wound is closed, again it will restore the basal lamina and also the interstitial matrix. In this way, what you can do is you can restore the dome shape, and dome shape was very much there. Only difference was they had extra tentacles, and this dome shape is restored now here, and it will start making the tentacles. So now if you look closely, the cellular dynamics, what is the cellular dynamics terminology means? That the movement of the cell. So no matter whether there is an injury or not, the Hydra body, the cells keep moving. They keep producing, or in other words, the Hydra body is constantly regenerating, whether or not there is a necessity for it. So cells undergo continuous proliferation and movement in both directions along the oral aboral axis. The empty cells denote proliferative cells, and the blue colored are the terminally differentiated one, as you can see in this picture. So the number of days taken for the cells to reach their respective destinations as buds or as hypostome. Hypostome is nothing but the mouth. And the peduncle termini are shown in the direction of red colored arrow direction. So the arrows depict the direction of cell movement. Like you see here, this is a tentacle, and the tentacles are terminally differentiated cell. That means they don't have a movement backwards. So from around the neck region, there is around 4 days it takes to reach the tip of the tentacle, and from the tip of the tentacle it will fall off. It is not going to stay back there, and constantly new cells keep coming. And like in the case of, you may have seen about the teeth, even for elephants and shark, new layers of teeth keep coming from the backward, and elephant also it keep getting just like a train from the back, it keep coming, unlike us. So the cells keep coming from this neck region towards the tip, and whereas in the hypostome region it takes around 20 days, whereas this long distance tentacle, it takes only 4 days. And reason you can guess it, because the mouth is minimally moving, tentacles are constantly moving, so wear and tear is very high, so it need to be repaired. And then you can see near the neck region there is little, hardly any movement, and you can assume this may be the production site. Either it will go towards the tentacle region or it will move towards the body region or the basal region, where it will come down, and in this case a new hydra is being formed, and that is called budding. And here it is trying to make another bud. So this is a special zone in which we call that spot as zone of budding. And towards the below this zone, that is basically the basal peduncle where the foot is attached, you can see the movement of cells is pretty low, and it takes around 20 days to travel, minimal days, somewhat like that of the hypostome. So extreme and hypostome and the basal region, the movement of cell is minimal, but in the body region, the movement is very high. You can assume that this could be because of the active movement around the tentacle region and also in the body region, unlike what you see in the hypostome and in the basal region. So the different cell movements that take place during the maintenance of Hydra body plan are mentioned in this picture. So now if you see, to study the gene expression, like in order to understand how Hydra is regenerating, we need to understand what are the genes that is contributing for this regeneration event. So for that, scientists use a lot of techniques. It can be RNA in situ hybridization or immunofluorescence. All depend on some chemical mechanism. That means if you have the antibody with an alkaline phosphatase enzyme and you put the substrate for that, BCIP NPT, it will give a colored reaction. So wherever there is a colored reaction, that indicates there was the antibody. And if the antibody was there, that indicates it's a substrate was there, and substrate is your subject of interest. So wherever the protein or the RNA was there, it will go and bind, and now you are detecting it using an antibody. So that is what you are seeing here. So the major head inducer of the hypostome organizer is a set of Wnt protein acting through the canonical beta-catenin pathway. So beta-catenin pathway is one of the most important signaling event that occurs in every organism, starting from a hair growth on your or a skin growth, or even your nail is growing back, or even you had a mosquito or an ant bite on your skin, it has to grow back. Everything requires Wnt signaling, and the Wnt signaling is occurring when the ligand Wnt binds onto the receptor frizzled, and this causes an activation of this receptor, because of which it will attract a component from the cytoplasm, that is disheveled. Disheveled was part of a destruction complex. Destructing what? Destructing the beta-catenin. So every cell, the beta-catenin is produced abundantly, and it is needed for the cell-cell communication, which we discussed in one of the earlier classes. But in any case, it has to be degraded. Otherwise, it will go to the nucleus and turn on pro-mitotic genes, and you don't want unwanted proliferation any given time. Because of this excess beta-catenin will be degraded by destruction complex. But if the Wnt is bound onto its receptor frizzled on the cell surface, it will recruit the disheveled, which is one of the major player of this destruction. As a result, the beta-catenin is no more degraded. It will go to the nucleus, and with the help of collaborators like TCF, it will turn on. So what you are seeing here is, for the growth of a new Hydra from the budding zone, definitely require the beta-catenin pathway. Wnt beta-catenin pathway means Wnt is the ligand and beta-catenin is the actual messenger that goes to the nucleus and bind to the DNA and turn on the gene expression events. So the Wnt expression during the Hydra budding is what you are seeing in this purple colored reaction, which is the detection of the Wnt proteins. And you can see here extreme end. Other parts there is no purple color, it is only the extreme end which is determining the head region. As you can see here in this picture also, you can see this is the mother Hydra and this is the daughter Hydra. Both of them have the Wnt signal expressed in the hypostome region. These Wnt proteins are seen in the apical end of the early bud, defining the hypostome region as the bud elongates from its source of production. If GSK3, which is one of the member of the destruction complex, like I told you, beta-catenin is degraded and disheveled is a protein that is recruited by the Wnt ligand bound frizzled. But if there are other methods, like can we think of blocking this degradation by some other method? Let the disheveled be there, let Wnt not be there, frizzled no need to come into picture. All you can do is, in a given cell, if you prevent the destruction event, and one of the major enzymes that contributes to this destruction of beta-catenin is the GSK3 beta glycogen synthase kinase 3. And this GSK3, if you block it, what will happen is the beta catenin is no more phosphorylated, because of which it will not be degraded. So if you prevent the GSK3 from functioning, you will stabilize beta catenin. Because beta catenin is not degraded, it will go to the nucleus. It will look as if there is Wnt signaling. Although there is no Wnt, there is no binding onto the frizzled, but what you did is you played something inside the cell by blocking the GSK3. So if your GSK3 is inhibited, throughout the body axis, ectopic tentacles form at all levels, and each piece of the trunk has the ability to stimulate the outgrowth of new heads. So this Hydra will be now with the head budding out from everywhere its body, because everywhere there is beta catenin is getting stabilized. So if you stabilize the beta catenin, you will end up getting multiple heads instead of one head. So this is what you get it. So what you understand, this signaling is very powerful, that is capable of driving the formation of multiple hydras from one organism itself. So now if you look into the head inhibition gradients, if you look into the head inhibition gradient, we already saw that the head has the ability to form a new head. So what you can do is, if you take a small piece near the head region from a donor hydra and put it in transplant into head region of another hydra. Earlier we saw if you put it towards the belly region, you will get a new head that is being formed. But here, if you put near the head region, you don't see a head that is being formed. What happened to it? If the same thing you put it towards the belly region, you will get a new head. But here, nothing happens. On the other hand, if you take the same thing and put it into another recipient hydra whose head is removed, then what will happen? Not only its lost head will appear, but also this new transplanted disc will also give rise to one new head. That means you will end up getting two-headed hydra. One is the replacement of the old head, the other is formation of new head. So what is the difference? The only difference is, in the first case, the recipient had a proper head, whereas the second case, the recipient had no head. You decapitated. Before it is regenerated, you transplanted the disc. So you end up getting two heads. So this gives you an idea. A existing head, functional head, prevents the formation of one more head. It do not want one more head. That means some inhibitors are being secreted. Same way, this we have seen in an intact host. Like if you take a graft, put it in the basal region. This you saw in the previous class. Towards the belly region you put it. No matter whether the host has a head or not, new head will be formed. So subhypostomal tissue does not generate a new head when placed close to the existing head. That is the top panel. In the B panel, what you see, subhypostomal tissue generates a head if the existing host head is removed. And third, the subhypostomal tissue generates a new head when placed away from the existing head. That means closer to the head, there is a zone of no response or some zone of surveillance from the existing head. It don't want one more head. It is just like in a lion pride. If one male lion is there, it don't want one more male lion to come, either from within the pride or coming from outside. So they will fight. So one pride need only one lion. Rarely two will be there, which will be mainly brothers. So the host head appears to make an inhibitor that prevents the grafted tissue from forming a secondary axis in the new head. The identity of the new head inhibitor remains to be unknown. It appears to be a labile substance which has half-life of only 2 to 3 hours. How do we know? If you decapitate, immediately you put the graft the tissue, it will not form a head. But you wait for some 2 hours or 3 hours, then you put it, new head will be formed. So that means the inhibitory signal is a volatile substance, or it can sublime easily into the atmosphere. So you end up getting a head only if you do for 2 to 3 hours. That means it's a short-lived molecule constantly sending across. And the same short-lived molecule, when it reaches towards the belly region, it diffuses off. Hence, belly region is most welcome for formation of new head anytime, provided you transplant with a new donor tissue from the head region. It is thought that the head activator, which is nothing but wind, we have seen it, and the head inhibitor, which we don't know what it is. We are assuming it is a flammable or evaporatable or sublimable substance, are both made in the hypostome. Both source has to be from the same place. But the head inhibitor gradient falls off more rapidly than that of the head activator. Head activator has a long life, That is why you took from a donor and put it into a recipient, still it is forming a head. That means the designation or the message to form a head is there in that donor tissue also. If it is a sublimable substance, it would have disappeared. The place where the head activator is uninhibited by the head inhibitor becomes the new budding zone. So in a spontaneously budding asexual reproducing zone, the inhibitor signal is minimal. So hence it has the potential to form a new head. So what prevents the bottom of the hydra from forming a new head? Normally you can think, oh, inhibitor is faded off from the bottom. So why don't head will start appearing from the base? It doesn't form from the base. It forms from a somewhere in the midway, lower. If you cut hydra into 3 pieces, piece 1, piece 2, piece 3, it will come between the piece 2 and piece 3 region. That means lower 2/3 region it forms. So you can see in this picture, you can see several small peptides could have been contributing to this. Do we have some evidence? Let us see. The bud location as a function of head and foot inhibition gradients, that is what is depicted here. As you can see in this picture, you can assume that there may be, if there is a head determinant, there will be a tail determinant also. In young adult hydras, the gradients of head and foot inhibitors appear to block the bud formation. There is a head gradient coming from the hypostome region and there is a foot gradient coming from the basal region. So somewhere in midway they get nullified. It won't be exact midway. It depends on the quality of the molecule what is inhibiting. So the foot gradient diminishes fast and head gradient diminishes little slow. As a result, you have somewhere two, that is what you can say, head inhibition and foot inhibition. There is a midway, as you can see in this red. In a young individual newly developed bud, it is pretty closer. As the young adult, it will become little bone change, and in budding adult, what happens? It is kind of neutralized out. So the both, there is a you can call it as like, many countries you will say the boundaries will have a no man's land. That means the middle of two countries doesn't belong to anyone. So we call it as a no man's land. Doesn't belong to that country or this country. So such a no man's land, the budding can happen. So that is roughly around at around 2/3 of the downward part. The trunk, when the levels of both inhibitors are minimal, this is where the buds form. Several small peptides have been found to activate food formation also. So the specification of cells as they migrate from the basal region through the body column may be mediated by the gradient of an enzyme, that is tyrosine kinase, and its name is Shingard. There are many enzymes are there. One of them is this. So now, if you look closely, the gene expression events, expression profile of Wnt and TGF-beta. TGF-beta also we learned about it in the liver regeneration, etc. Superfamily signaling components along the Hydra body axis. So in this left panel, what you are seeing is the Wnt signaling events, and in the right panel, you are seeing the TGF-beta signaling. And what you see here is the molecular components that have been shown to be regulated by Wnt and the TGF-beta signaling pathway. So many genes are there. Let us don't focus about these genes. The colored bars represent the localization of gene expression along the body column of the Hydra polyp. That is what, there's a cartoon of Hydra that has been given, and if it is been shown here, that means up to here it is expressed, and if it is shown only here near this narrow window, it is expressed only here. If this one, green one, is shown up to here, and green basically means agonist of the signaling pathway, blue means antagonist. Like when you say Wnt signaling, there is agonist and antagonist. Like Wnt ligand is an agonist, and there are many inhibitors are there for Wnt signaling. SFRP is there, DKK is there, WIF is there, many other molecules are there. They are all antagonist of Wnt signaling. So these molecules, what you see, they are either agonist or antagonist. And TGF-beta signaling also, you have agonist or antagonist are there, and you see the expression pattern. Like some starting from the neck region, say this particular thing, until around the foot region it is expressed. And whereas some genes, like this particular gene, it's expressed very closely to the hypostome region. And you can see the blue color is the antagonist. And you can also see this terminology is mentioned here, ectoderm or endoderm. Like you have a whole column has got both ectoderm and endoderm, and its EC means ectoderm, EN means endoderm. So that is what has been mentioned here. Location is not important. This data is mainly present to make you understand that so many pathways, here only 2 pathways are been looked into, Wnt and TGF-beta signaling and their agonist and antagonist. It is a complex event, gene expression or the orchestration of event that do happen. So the colored bars represent the localization of gene expression that you should understand here, and the expression of genes are ectoderm or endoderm also specifically mentioned, both for TGF-beta signaling and Wnt signaling. So the Hydra body plan is characterized by a defined body plan that is consisting of stem cells, a simple nervous system, and 2 differentiated tissue layer, that is ectoderm and endoderm, and in the middle you have a mesoglia and also gametes, nerve cells, or some nematocytes, etc. So the Hydra consists of more than 3 active but separated stem cell communities: the ectodermal epithelial stem cells, endodermal epithelial stem cells, and interstitial stem cell lineage. So these are all the main stem cell lineages. In Hydra, all 3 stem cell lineages is highly dynamic, and all cells are continuously renewed every 20 to 30 days. This is something, a unique feature what you can see in Hydra. This continuous proliferation of epithelial and interstitial stem cells occurs in the central part of the gastric body form, from which the cells migrate either to the upper or to the lower. This we saw in the previous picture also, upper or lower body column, or towards a growing bud. If bud is growing, then there will be more migration towards the bud as a part of the asexual reproduction. At both ends, unipotent epithelial stem cells differentiate into head and foot somatic tissue, thereby losing the potential to initiate the cell division. So multipotent interstitial stem cells differentiate not only into non-dividing somatic cells, for example, nematocytes and nerve cells, but also into gametes, that is eggs and sperm. So this is the steady state way of movement and migration of the cells in the body of Hydra. You can see here, this has been depicted in the form of a cartoon. You have got endoderm, mesoglia and ectoderm, and you can see the cells that is located, and the ectodermal and endodermal epithelial stem cells, they keep cycling around 3 to 4 days, and they can get differentiated into somatic ectodermal and endodermal tentacle or basal disc cells. And you can see this is a stem cell and this is a precursor cell, and here in the bottom you can see 4 classes of differentiated somatic cell. The nature of these cells are not important. It has been shown mainly to understand that the newly formed Hydra, as a part of regeneration, it keeps producing every cell that is present in the organism. Gland and mucous cells, ganglion and sensory neurons, and nematocytes, germline cells, etc. And this is an infinite cell division that constantly keeps going, as long as, because Hydra cannot die, it constantly keep producing new and new, just like bacteria. Bacteria normally don't die because they keep producing, one bacteria keep producing 2 bacteria, 4, like that. Differentiation into the somatic cells happen, and eventually the cells will die, and it will fall off from the tip of the extremities of the body, but it doesn't matter, constantly it keeps pushing forward. Let us look into few other molecular mechanisms. Although we have seen about the importance of TGF-beta and Wnt signaling, regeneration starts with a wound. The healing may require some special action not essential during normal morphogenesis. Normal morphogenesis, all you see is cells keep dividing, stem cells keep replacing the newly formed cell, and they keep moving across the body of the animal. That is the normal morphogenesis. An evolutionarily conserved gene called Cell gene, which is also known as SPINK3 in the case of human, which is expressed. Remember, this Hydra gene is present up to human. That means how conserved this gene. That is why Cell gene is very important. It is basically a serine protease inhibitor, Cell type. That is what the abbreviation of SPINK. And in Hydra it is called Cell, which is expressed in the endodermal gland cells. Inside the body, endodermal gland cells, and upregulated during regeneration. Cell1. In human, the same gene is called SPINK3. Cell1 silencing by RNA interference resulted in dramatic tissue disorganization. followed by a massive death of gland cells and the accumulation of autophagosome. Autophagosomes are bodies that is formed inside a cell when the cell is killing itself. That is called autophagy. That means eating yourself. Something like, if you are hungry, you will not eat your finger, right? If you are hungry, will you start eating your flesh in your hand? You don't. Normally you don't. But the cells can eat, and when it is not finding a way across, way forward, it will undergo autophagy due to various biological conditions, and it will recycle the nutrients. So autophagosomes are required for this autophagy within the cytoplasm of the digestive cells. So what you did was you got rid of KZL1. So that will trigger the self-destruction of these cells. Intact Hydra, KZL1, which is a serine protease inhibitor activity, is required to prevent excessive autophagy. If KZL is not there, you will have excess of autophagy. So KZL1 makes sure that you don't have serine protease. Protease means a enzyme that is degrading various proteins. So that is kept in check. It's just like you keep petrol or away from fire, or fire away from petrol. Same logic applies. Serine protease has to be kept in check, which is done by the KZL1. But once you block it, it goes unchecked, and various serine proteases will start acting to prevent excessive autophagy and to exert a cytoprotective function to survive the wounding stress. That is why the sudden increase in KZL1 is necessary for the regeneration event to happen, and this gene is PINK3 in the case of human, and that means it is present in all other animals also. How do the cells in the regenerating tissue know? How do they know? That means it's aware of it, that their localization in the gastric region has been changed to a position at the apical or basal end. Are they looking into the mirror to know that, oh, my head is missing? Or is someone is telling, oh, head is, do they have any news channel that is reporting the head is missing? So this is what we should know, that what part is damaged. Secreted peptides are to be responsible for this, have an impact on the head and foot regeneration. Several peptides could be linked with epithelial differentiation along the apical basal body axis, capable to induce head or foot specific differentiation. A 12 amino acid peptide called HEDI is a novel gene that is absent in the genomes of other animals. That is, 12 amino acid peptide HEDI, HEADY, is a potent inducer of apical fate and also sufficient for head induction. So the HEDI peptide, if you deliver in a place, it can give signal to that cell to start making a head. So disruption of HEDI function by double-stranded RNA mediated interference, RNAi, which is a technique that has received a Nobel Prize, resulted in a severe defects in the head formation. So if you get rid of HEDI, that is a 12 amino acid peptide, you will not have head formation. Treatment of regenerating heads with synthetic HYM301 peptide that causes increase in the number of tentacles formed. One gene, that is a synthetic HYM301, which is a gene, if you treat them, a more number of tentacles are formed, while the treatment with HYM301 double-stranded RNA, that means you are knocking down this HYM301, leads to a reduction in the number of tentacles formed during the bud formation or the head regeneration. At the opposite end of the body axis, two peptides, that is pedibin and pedin. Pedin's another name is HYM346, stimulates foot regeneration. HYM346 accelerate foot regeneration and also increase foot activation potential in the gastric tissue. So you can see in this picture the HEDI expression and HYM301 expression that is been shown here. Like you can see at different time points, this is the decapitation, and you can see soon after that at various time points, 2 hour, 4 hour, 6 hour, 8 hour, you collect it. You can see the HEDI induction happens strong at around 6 hours and then it dwindles off. And this is the expression of HYM301. This is the expression of HEDI. And the HEDI expression is very specific in the neck region and also in the newly budding region. So we will continue with some of the mechanisms in the next class also. And also we will study more about how Hydra is using this for morphallaxis and other asexual reproduction approaches. Thank you.